Okay, so we're going to spend this week talking about cancer, and I'm hoping that we're going to then knit together a lot of the ideas that we went over during the first 4 weeks, and you'll see how they all fit together in an understanding of this disease. So this is the second year where I've dedicated this to Greg, and I want to tell you a little bit about Greg. So Greg was someone I hired as my lab manager 3 years ago this June. And he was essentially the same age as all of you. He was just hired out of, basically just out of undergraduate. He was incredibly excited to get the job. I picked him out of a bunch of people, and it was exactly what he wanted to do. This is what he wanted to do, his research. And this was his first career job, and so he was really thrilled to do it. And so he started for the first 3 weeks, and he was getting everything figured out, getting a lot going. And after 3 weeks in the lab, he started developing this persistent headache right behind his eye, which he thought had something to do with his contact lenses. And he went in to get it checked out, and in short order, they figured out it was a glioblastoma, which is a type of brain cancer. And so they immediately treated him. He went in for surgery, chemotherapy, radiation, the whole bit. And I didn't see him for 6 months. He came back after 6 months. He was very weak, but he thought he could get back into it. He worked for a week and it relapsed, and he died 7 months after he started. So, and he was 23 years old. So it was a very sad situation. And so we talk about the biology of the cancer, and we'll talk about the medical implications, but it's a very real thing, and it has very real implications. And the thing about cancer that makes it hard is that the number one killer is heart disease, which is, and you can sort of look at it and say, well, maybe if people hadn't eaten so much hamburger and steak, they might not have gotten it. But there was nothing Greg did wrong. He was very healthy. He was in good shape. He was just very, very unlucky. And that's the thing about cancer. It's really just a matter, in many ways, it's a matter of luck. There are certain things like, if you smoke 2 packs a day, yes, there's a very high chance of getting lung cancer. But for most of us, it's really just a matter of luck, good luck and bad luck. And Greg, sadly, was very unlucky. So in any event, I want to begin with an analogy. It's kind of a strange one, but bear with me for a few minutes. So I want to talk about Syria. You probably know something about Syria. I would think so. It's been in the news quite a bit lately. It's been a sort of protracted problem. And the thing about Syria is it's actually the size of the country is almost exactly the size of Washington State. It seems somehow much bigger when you read it in the news, but it's actually not any bigger than our own state. The population is about 2.5 times. And the thing about Syria is that it has a lot, it actually goes way back in history. It's an extremely old civilization and has all these different kinds of groups there. There are Sunnis, Shiites, Christians, Kurds, and they basically all live together. Sometimes they had their own communities, but especially in the bigger cities, they would often intermix and live together. And it was certainly no picnic living there. It's been under a brutal leadership, but people got along. And then starting in 2011, things started going really bad. There was first the Arab Spring, which you may have known about, where there was a sort of revolution that was sort of was brutally crushed by Assad, the dictator, and then ISIS came in and basically started taking over territory. So just to show you, this was one of the major cities, Aleppo, 2011. This is a mosque. This is what it looked like 2 years later. And you can just see, picture after picture of Syria like this, just complete destruction. And so, okay, so you wonder, well, where am I going with this analogy? Well, the thing is about cancer, it's kind of like that we have over 200 different cell types in our body. And the way we exist as organisms is that all those cells have to respect their own territory. As long as they all work together, they intermix, they respect where they are, everything goes fine. And what happens in cancer, it's actually a lot like ISIS. It comes in and it just doesn't care about anything else, and it's just destruction until eventually it'll, as often happens the case, kill the organism. So that's the sort of first thing, is that it's this change from this ability of the cells in our body to kind of work together to having this one cell that really doesn't care about the health of the organism, simply wants to proliferate. The second reason I bring up this analogy is if you look at Syria, it's really easy to pick one thing and say, oh, it's all ISIS, but that clearly wasn't the case. There were many, many causes that led to Syria being where it is. For example, the fact that there was a brutal dictator that people really hated, that's why there was a revolution in the first place that gave an opening for ISIS to enter. The fact that, the US and the Russians and Iran all came in and saw, wanted to push their own agendas and so on and so forth. And this is also what I really want you to think about when you think about cancer. It's not like just one thing goes wrong. It's not like that one cigarette you smoked in high school or that one thing you ate that caused all the problems. Cancer, as we'll see, is a multifactorial, has multifactorial causes, that there's a lot of things that have to go wrong in order for it to happen. And so I want you to keep those ideas in mind as we go through all these different aspects of cancer. Now, I don't expect you to memorize this by any means. I just wanted to give you some perspective, though, on cancer, the number of deaths. This is just in the US. It's slightly old, but it's more or less relevant. And this doesn't include the most common form of cancer, which is the skin cancer, the non-melanoma form of skin cancer. And we'll talk about that one later. And that's because it's rarely lethal. But you can see there's on the order of, over 1.5 million new cases a year, and over, almost 600,000 people a year die of it. So it's the second biggest cause of death in the US. But what I also want you to see is that cancers, and we'll stress this more and more, are very different in their biology and their underlying causes, and even in their how lethal they are. So if you look at colon cancer, about half the people who get it more or less will die. Pancreatic cancer, by far the worst one, that's the one that killed Steve Jobs, has a very, if you look at the number of new cases versus number of deaths, you can see the odds of surviving it are not good. Same for lung cancer, the most preventable of all the cancers. Whereas others like prostate and breast cancer, the odds are much better. So the cancers are really different, and just because you get cancer doesn't mean it's a death sentence by any means, but it certainly depends on the type of cancer. Now, the other thing about cancer is that it's really a matter of age, and I said it's sort of a matter of luck, right? But the thing is we'll see is that as you get older, the number of sort of bad luck things that can happen increase. So the older you live, the more and more your chances of cancer increase. That's why, they talk about these people having these ideas they're going to they'll extend their lifespan by this and that. This is always one of the big issues, that the longer you live, the higher your chances of getting cancer, and we'll see why that is. So let's go back to where does cancer come from, and let me just, you've probably heard quite a bit about stem cells, so let me just remind you, though, that we have a lot of different stem cells. We'll talk some about them specifically, but basically a stem cell will divide, and in general, one will give rise to a daughter cell that will basically retain the original stem cell. That's the key thing, that we retain this population. And one will go on to terminally differentiate. Now, as my little note on the side indicates, that this 50-50 is true as a population, but it's not rigorously true. So a stem cell can give rise to two daughter stem cells, it can give rise to two differentiated cells, or it can give rise to 50-50. It's all sort of regulated so that on average this is what happens, but it's not strictly determined to be 50-50 for any single division of a stem cell. Now, even when the stem cell divides and one of the daughters commits to differentiation, it's not that it immediately differentiates, but instead it undergoes this amplification. It's called the transit amplifying. So one of the daughters, once it makes the decision to differentiate, then undergoes these series of very rapid divisions, so that when it actually differentiates, there's actually a lot of daughters that came from a single stem cell. So each stem cell will give rise to lots of differentiated cells. So let's look at a couple examples. Here's one, the skin. You remember we talked about the skin when we talked about EBS. talked about how the dermis is down here. And you remember I told you about that extracellular matrix, that sheet of extracellular matrix. That was the one that was disrupted in that boy who had lost most of his skin. That sits right there. And I told you that on top of that are the actual stem cells. Those are the ones where you can have mutant keratins in EBS. And what happens is these cells slowly divide in what's called the basal layer. There's a cell dividing right there. One of the cells will stay behind on average, one of which will begin the differentiation. So it'll go through this transit amplifying phase where it proliferates a lot. It moves up into these upper layers as it proliferates and differentiates. And eventually it goes through a series of changes where it loses the nucleus and the cell dies intentionally and forms this hard outer layer. So this is the outer layer of our skin. And this is constantly, the cells are going through this process. So from the time a cell is born till it sloughs off is about 3 weeks. And one of the disgusting but true facts is, you know, all that dust you have around your house, well, most of that is either skin you shed or your roommate's skin. That is because we're constantly sort of peeling off skin. We don't notice it, but that's what's constantly happening. And that's for a good reason, because the outer layer is subject to all sorts of damages we'll talk about later. And this protects our stem cells, the underlying cells, from damage. So most of the time, any damage, any nasty things we're exposed to only hits the outer surface. Now, another example we'll talk about is in the gut, in the colon. And we have, this is just a little projection of what it looks like. We have all these little things sticking out into our gut that secretes all the enzymes we use to digest our food. All sorts of hydrolases, proteases, and so forth. And so you see these little things sticking up. They're called villi, and they're continuous with these things they're hiding down in here, which are called the crypts. Now, if we zoom in on a crypt down here, what we'd see is down near the bottom are these stem cells that slowly divide. So the stem cells divide like once every 24 hours. One of the daughters stays behind on average. One of them differentiates rapidly. So you go through this rapid cell division phase. And then after a time, it stops dividing and begins to terminally differentiate. But because new daughters are being born here, the cells are constantly being pushed up over and over again. And so eventually they get to the top and shed off. And so the time from when it's born to when it sheds off is about 3 to 5 days. So if you suddenly stopped all your stem cells dividing, within a few days, you basically would have no lining of your gut because all the cells would be dead. So we need this. So why do we have this? Well, because you're eating all sorts of crap in the food you eat, like that broiled hamburger you had with God knows what's in it. And it's going in here and all that stuff, all those nasty carcinogens are sitting in there. But the cells they're affecting are only the terminally differentiated cells, which are going to die anyway. So if they get mutant, it's not really a big deal. Whereas most of the time, sitting down here, your stem cells are protected. There's actually tight junctions in here that prevent your stem cells from being exposed to all the stuff in your diet. Okay, so again, it's this theme of keeping the stem cells buried and having differentiated cells exposed to all the nastiness because they're going to die off soon anyway. So let me just show you a little movie about the colon.
The lining of the small intestine, like the lining of most of the gut, is a single-layered epithelium. Depending upon location in the gut, the epithelial cells, usually called enterocytes, help absorb nutrients from the lumen of the gut, or absorb water from the intestinal contents. In the small intestine, the surface of the lining of the gut is increased enormously by thousands of villi that protrude into the lumen. Here we see a single villus and its internal architecture. The surface of the villus is covered by a single layer of enterocytes, which extend down into the crypt below. Stem cells at the bottom of the crypt located between Paneth cells, divide and make copies of themselves, and also make transit-amplifying cells. The transit-amplifying cells proliferate rapidly and move up the walls of the crypt. As the cells migrate upwards, they begin to differentiate into goblet cells and enterocytes. While the cells are moving up the sides of the villus, they carry out the essential functions of the small intestine, notably absorption of nutrients. When the differentiated cells reach the tip of the villus, they undergo apoptosis and are shed into the lumen of the small intestine. The entire process of outmigration and cell death is completed in just 3 to 4 days. The process of outmigration and rapid cell replacement is a defense mechanism against the development of colon cancer, since almost all epithelial cells, including those that have accidentally sustained mutations, are shed within days of their formation. Therefore, the only mutations that can lead to the development of a cancer are those that are retained in the crypt. This dictates that such mutations must block the outmigration of mutant cells from the crypt.
Okay, we'll leave it there for now. So we're going to be talking about both skin cancer and colon cancer. That's why I wanted to introduce these here. But first, we're going to talk about sort of, as I said, every cancer is different. You can't talk about cancer as one thing, even if that's sort of how it often seems from the popular press. But we'll really see that how different cancers are. And we're going to get more and more detailed about this because I think it has a lot to do with what the future of medicine is about with regard to cancer. So, so one of the things is that you don't just suddenly get cancer. It doesn't just come out of nowhere. There's actually a series of stages that change, and I think the one we can see it most beautifully in is in the stages of uterine cancer. So this is the inside of the uterus. So here is the underlying tissue. There's a mat of basement membrane right there. Sitting on top of it, just like the skin, is a set of stem cells. The cells will, again, give rise to daughter cells, the transit-amplifying cells that undergo a number of divisions, and then here's the lining of the uterus up here. And what can happen initially is there can be an overgrowth of some of these stem cells, transit-amplifying cells. You can see there's sort of a darker color with this nuclear stain, and you kind of get a bit of a buildup here. The oncologists will call it a low-grade neoplasia. So there's a bit of a buildup, but you can still see they're differentiating to give rise to the outer layers. If it progresses further, it can get to what's called a high-grade neoplasia, where what you can see is that you can see how all the cells are the sort of transit-amplifying or stem cells. And this isn't like covering the whole uterus. This is just going to be one little spot. So the good news is if this is caught, what can happen is it can be just surgically removed, and the good cells will come in, the good stem cells will come in from either side and fill it in. The bad news, and we'll talk about this on particularly more on Friday, is that if the cells then invade across the basement membrane, which, as I told you, plays this very important role in keeping the epithelial cells from the underlying tissue. It protects us in many ways from cancer because these cells are confined here where they can be removed. But if they develop the ability then to invade across the basement membrane, they can then spread through the underlying tissue and ultimately spread throughout the body, the process we know as metastasis. Okay, so this is really bad. So you really, the key thing is you really would like to get a cancer in this stage. And so, you know, detection is obviously a key thing. So this is one of the ways, or the main way to catch it in terms of uterine cancer is a Pap smear. And what they do is they come in and they scrape a little bit of cells. And if if there's a little scrape, and the cells up here, you can see these epithelial cells. Normal epithelial cells have very distinctive morphology, very flattened cells with these little nuclei. So that would be good. That's the news you want to get. However, when they do the scraping, if the pathologist sees within there a mixture, you can see there are some normal cells there, but they're also detecting these cells, larger nuclei, much smaller cytoplasm. Those are very indicative of these cells in this case. And so if this is caught early enough, then again, they'll come in and they can surgically remove the problem. This is what, when it goes on to the next stage, invasive, it's hard to see in these pictures, but the cells begin, remember we talked about lamellipodia and filopodia. Invasive cells have, as we'll talk about later, have all these protrusions on them. And it's bad news when they pick up cells like that. Okay, so again, it goes through stages, and we'll go through that again in a second. So let's just talk about some general properties of cancer. So one of the things is it's basically an overproliferation of stem-like or transit-like cells and a failure to differentiate. So again, this is the normal process. You have stem cells. They'll divide a fixed number of times depending on the cell type, and then they'll terminally differentiate. And what happens in cancer is that you get this overgrowth. This is meant to indicate some sort of mixture of mutant stem cells in red as well as the transit amplifying cells. So the tumor is often a mixture of these. And this is just to indicate that the way people believe it works is that what can also happen is that, particularly in metastasis, stem cells can then break off, migrate, and then create new cancers. That's how you get sites of new cancers in the body as derivatives from original stem cells. But the main point I want to emphasize is that there's an overgrowth of these types of cells. The second thing is that cancer, you can trace it back, and we'll see more of this in just a second, to a single cell that grows a lot. So what can happen, this would be, say, the early progenitor of the cells that are going to give rise to the cancer, one cell way back in time. And then the tumor can first be seen on an X-ray at 10 to the 8th cells. It can first be felt, that's the word palpable, at 10 to the 9th cells. And, and again, don't take these numbers with hard and fast, but when it gets up to about this number is when it starts killing people. So the point though is that there's a lot of divisions. That's why even when people show up in the clinic with, and they're first recognized to have cancer, the originating event could have happened many, many years ago, depending on with a fast-growing cancer more recently, some very slow-growing cancers, it could have happened a decade before. As I talked about in my analogy, cancer begins with a single cell that undergoes mutations. This is sort of, we'll see a more realistic example of this, but this is just a sort of schematic idea from Albert's. You have a set of stem cells. One cell picks up a mutation. We'll see it's not just one, but picks up a mutation. And then, it begins to grow a bit too much, so the mutant stem cell grows a little too much, but that's no big deal. It's confined to the epithelia. Then there's a chance for a second mutation. It begins to grow up too much. You begin to get those neoplasias. But again, it's not such a big deal because this can be surgically removed. But the bad problem is if you pick up additional mutations, that's what can lead to the breaking through the basal lamina and ultimately the metastasis. So again, the idea, it's not just one thing. It's not that one cigarette you had that leads to lung cancer. It's repeated mutations. So this was sort of the general idea. There were a few mutations behind cancer. But with the advent of sequencing, we've learned, in fact, something really shocking. And this is from the original publication. Alberts has a version of this, but I'll just show you the original one. And what we've learned is that people have enormous numbers of mutations. So this is from a woman who had breast cancer. The breast cancer was biopsied somewhere along in here. And when they biopsied it, they discovered that she had 71,000 mutations in that biopsy. 71,000, okay? Not 2, 3, or 4, huge numbers. So the authors who did it, there are a number of things they recognized that I think were really interesting. One is that even what we think of is that tumor isn't a single thing. It's actually composed of different populations, and they can recognize this by doing single cell sequencing. So it's composed of different mixed populations. And this is part of the problem of cancer, is that if you look at these different populations, if you come up with a treatment, you may get rid of like almost all of these things, whatever treatment you come up with. But let's say the blue one, which could actually be maybe only 1% or 0.1%, maybe that doesn't get killed. So someone goes through treatment and they think they're completely, it's all cured, they can't detect any more sign. But that small percent that is left over can over years then hide and eventually start growing again. And that's what often leads to a relapse. So it's one of the really hard things about cancer. So the other thing is that this is all completely fanciful, but they sort of, they made an idea of what was happening during her life. So somewhere early in her life, we don't know where, maybe 10 years before, she started picking up a mutation. Maybe this was the first mutation up here somewhere. And, it didn't really have an effect. And, it was just that one cell maybe picked up some mutations. But then somewhere along the line, maybe up in here, God knows, they're just guessing all this stuff. But maybe after a year, that began to expand a bit. Those stem cells, they began to overproliferate in like that low-grade neoplasia. And things kept changing and changing. Maybe it split into multiple populations. Somewhere in here, you begin to get chromosomal instability, which is that basically the chromosomes, this happens a lot in cancer, undergo large-scale mutations. And once the chromosomes start undergoing instability, more and more mutations begin to pile up. Somewhere, they guess maybe around 50% of the time towards the development of the cancer, they could find one mutation that was present in all the populations that eventually survived. So this was sort of the original common mutation. But you can see that the idea is that there were different populations. Some maybe arose and then died off. So it's sort of like this ever-changing thing. It's sort of like the war in Syria. ISIS kind of shows up and then it goes back, and the Nusra Front shows up and da-da-da. And it could be like that certain populations grow and then die off. But eventually, we get this full-grown effect. And so this is when she came into the clinic and they biopsied it. So I think it shows you the challenge that one faces in dealing with cancer, that it's not, if it was just one mutation, you could come up with a very easy or relatively easy cure for it, but because there's so many, it makes it a challenge. So this is a theme that we'll look at as we go on. Now, as I talked about with Greg, cancer is often bad luck. This is meant to be a slot machine pulling up 3 whatever X's, okay? And it really is like that. It's just a lot of it's just bad luck. It really is bad luck. Say Greg was healthy, good shape. There was no reason he should have gotten cancer. And one of the things that's sort of come out more recently is people believe a lot of it is just due to inborn errors of replication. Every time our cell divides, and our cells divide a lot, there's always some chance of DNA replication errors. We have ways of fixing that, but they accumulate over and over time. Most of them are completely innocuous, but as we'll see, some of them may not be innocuous. And if you get enough of the mutations in the same cell, and it has to be in the same cell, then it can go on under the right conditions to go on and form cancer. So you could live the most perfectly healthy vegan lifestyle living up in the Rockies where you never look at the internet or God knows what, and you could still get cancer. It just, and not to say that there are things you can do. You can certainly increase your odds of getting bad luck, and we'll talk about that. But everyone has an odds of it, and so it's not like someone like Greg did anything in his life wrong. Now, part of it is due to genetic complications, errors, but the other thing that also makes it complicated is their environmental contributions too, most of which we have a very hard time understanding. And this is just to show you, this is a picture from Alberts again, showing you that one of the ways we know it is that when people go from one place to another, types of cancers in those people can increase or decrease. So for example, if you look at people who go from Japan to Hawaii, they have a decrease in stomach cancer, but an increase in prostate, colon, and breast cancer. And genetically, they're all the same. They're people from Japan, but it must be something about changing from the Japanese diet to the Western diet increases some kind of cancers, decreases others. And that's why in the popular press, we always see these articles on, like on page 1, eat this and you'll avoid this cancer, eat that and you might get that cancer. But it's and then it changes, a lot of times, because it's very hard to do these epidemiological studies. Clearly there's a contribution though, but except for a lot of cases like asbestos or cigarettes, we often don't know what it is, and they're often very subtle effects. But clearly the environment is playing an effect. Okay, so that's sort of the background to cancer in terms of sort of the larger picture. Now what I want to do is start talking about what actually causes cancer. What are the sort of, at a molecular basis, can we understand about that? So there's two kinds of genes that are involved in causing cancer. And the first one we call proto-oncogenes. These are genes that cause cancer when they're activated. And sometimes people come to my office hours and say, well, why do we have these genes in us that cause cancer? And what I want to emphasize is these are normal genes. It's just that some of our genes, when mutated, can lead to cancer. And we'll see a lot of examples of that. And there's a lot of different ways these particular class of genes, and there are many of them, thousands, can be activated. So you can have, let's say, a protein that is normal, but it picks up maybe a point mutation, some sort of mutation that converts it from a normal state to a cancer-causing or oncogenic state. Another case can be that the protein itself doesn't actually undergo any change, but it's just that when there's too much of it, it can lead to cancer. Or in other cases, it might be absolutely fine when it's expressed in the oval cell, some sort of cell type in your body, the oval cell, but when it accidentally gets expressed in the round cell, some cell it's not normally in, it leads to cancer. So different kinds of changes. And there's a variety of different things that can happen. For example, there can be a change in the coding sequence, a deletion, a point mutation that can cause the protein to be either abnormal in function or made in abnormal amounts. There can be mutations in the promoters, so there's too much of it produced. There can be what's called gene amplification, where because of chromosome instability, the same gene is produced in many copies, leading to too much of it. Or, and we'll look at a case of this, there's a thing called chromosome rearrangement, where different pieces, because of this instability, come together. So a strong promoter is put in front of a gene that's normally expressed weakly, causing too much of it to be made, or a fusion of two different coding regions come together to produce some sort of hyperactive protein. And I've also added to this, leading back to the very beginning of our class, another thing that people have now recognized to be a cause of cancer is a loss of a microRNA that would normally keep the levels of a protein down. When the microRNA is lost, then we get too much of it. So there's a good news, bad news scenario with proto-oncogenes. So the good news is, even though there's all these different mutations, the odds of any mutation causing something to be activated is quite low. Oh, sorry, just yell if I don't respond.
In this case, it's about proteins being made too much, right? Not about like cell proliferation or...
Well, we'll see, yes. But the initial, you're right, proliferation, as we'll see in a minute, is the downstream cause, but the initial cause is the protein itself being made too much, right? For one reason, or being hyperactive for some reason, right? So exactly. So the odds, these things don't happen very often. Lots of mutations can happen to a protein, for example, but one that causes it to be hyperactive is a very rare mutation. So the good news about proto-oncogenes is the mutations themselves are rare. The bad news is it only has to happen to one of your two genes in order to get cancer. Now, the second major class we're going to talk about are what are called tumor suppressors. And these are proteins within us that work to limit how much cells can grow, just like I mentioned a minute ago. They're going to regulate how much cells grow. And we'll go through examples of all these. So this is just giving you a flavor for them. But what happens with tumor suppressors, unlike proto-oncogenes, is they lead to cancer when they're inactivated, when they're turned off. Now, let's just look at a case of, here, this is one from Albert's, just an example called retinoblastoma. As we'll see, it actually is involved in many cancers, but it was originally discovered in eye cancer, hence the name, the retinoblastoma gene. But basically, what can happen to a lot of us is that in our cells, we get a mutation, so one of our two RB genes is inactive in a cell. Well, it turns out that's not a problem because we have two of them, right? So you lose one, the other one's going to be perfectly fine. You'd never even know you had the mutation. Probably in each of you probably have 1 million cells. Each of us has 1 million cells in us that has a mutant RB gene. We have no idea that's going on. Not a problem. The problem is that if you get unlucky, again, it's bad luck, and in the same cell, the second gene gets knocked out. The same cell. So the odds are pretty unlikely. I mean, it's not unlikely to get a knocked out gene, but to get the same gene knocked out in the same cell is pretty unlikely. So that's rare. What is much more common, and we'll talk about this in more detail, is that people can be born with a mutation in one of their two genes. This is not uncommon, and we'll talk about very specific examples of that. But if they're born with a mutation, then it's much more likely they'll pick up a second gene. So for example, retinoblastoma is really rare, but if you're born with a mutation in one retinoblastoma gene, the chances that you get eye cancer go up tremendously. Okay. Now, the reason that you can lose the second gene is from a variety of things. A point mutation that, for example, introduces a stop codon is a very example one. Deletion of the gene or a part of the gene. Epigenetic silencing, right, that turns off the genes. Or, again, coming back to what we talked about earlier, a microRNA that normally isn't produced gets cranked up to high levels and shuts off the gene. So with tumor suppressors, there's also good news, bad news. So the good news, as I told you, is the odds that you're going to have both of them knocked out by whatever means in the same cell are pretty rare. Okay, that's unlikely. The bad news is it's pretty easy to knock out a gene. A lot of mutations can knock out a gene. So again, it's as with proto-oncogenes, good news, bad news situation. So we'll talk about, we're going to get into the weeds about what proto-oncogenes and tumor suppressors are. Let me just tell you though that a big, and I sort of alluded to this earlier, that a big part of cancer, and this comes back to your question, is an overgrowth of cells. So cells growth is highly regulated, right? It's extremely regulated. That's why I spend so much time on the cell cycle. It's very tightly regulated. So for example, cells will not grow when they don't see, in general, when they don't see something called a mitogen, something that causes them, it comes from the word of going through mitosis. But when they see a mitogen, a signal, that will tell them to grow. So what a mitogen is, there's lots of different mitogens, is that they're basically signals that impact on the cell that work through things like second messengers and kinase cascades, all the stuff we spent time talking about, that lead to cell division. So when that signal is there, the cell knows it's ordered to grow. When the signal isn't there, it's time not to grow. And we'll talk about more about mitogens as we go on. Now, I just want to give you a little bit of terminology. So technically, a mitogen is something that causes a cell to go through a cell division, okay, to actually go through the cell cycle, and we'll see this. A growth factor, technically, is something that causes a cell to increase its biosynthetic activity, which, as we talked about, is necessary for a cell to divide. So you need to drive through the cell cycle, but it needs to actually grow. The complication in the real world is that a lot of things that we call growth factors and many signals actually regulate cell division and growth. So the term, like if I say epidermal growth factor, don't think it means it only affects cell growth, because it can affect mitosis as well. This is sort of the terminology we're stuck with. But technically, you should know this, a mitogen is something that causes cell division, growth factor is something that causes a cell to grow. So any of these signals. Now, there are many other things that impinge on the decision to grow or not grow. Another is how cells are packed against each other. So when cells are in contact with each other, they often don't grow. But when they're sparse, they know they're supposed to grow. And an example of that is if you think about your hand and you get a cut, what will happen is the cells that are normally, the stem cells that are nice tight against each other, when you have a cut, they're separated. That tells the stem cells on either side of the cut, well, wait a minute, there's nobody on either side of me, and that'll be a signal, so they'll grow in, and that's how we repair the wound, okay? That's part of the triggering. So the cells not being in contact is an important signal, and we call it contact inhibition.
When normal cells are introduced into a petri dish at low numbers, they begin to grow and divide. As the cells begin to touch one another, they stop dividing. This response is called contact inhibition. Once the cells fill up the bottom of the dish, all cell division ends, creating a state called confluence. Contact inhibition ensures that the cells create a layer only one cell thick, a monolayer. The behavior of cancer cells is quite different. If a cancer cell is seated among normal cells, all of the cells will proliferate as before. However, once confluence is reached, the normal cells will stop multiplying, while the cancer cells continue to divide, yielding a clump of cells, often called a focus. Contact inhibition can be demonstrated in vitro by removing cells from a confluent monolayer. In this experiment, cells are removed by scratching the monolayer with a needle. The surviving cells at the edge of the wound now do two things. First, they begin to proliferate again, since they are no longer fully contact inhibited. And second, they migrate into the empty area of the wound, attempting to fill it up.
So that's exactly what happens when you get a cut. It's the same thing. You get proliferation and cells moving in to fill up the wound. Okay, so now let's come back to the cell cycle. So remember when I told you about the cell cycle, we talked about the different phases, and one of the things I stressed was that G1 and G2 were not resting phases. They were actually activity growth periods. But cells do have a resting period. It's called G0. Actually, you may notice I moved it up here, which is sort of, it used to always be traditionally placed down here, but there's new evidence that puts it more up here. But regardless, it's a resting period. So what will happen is cells can sit in this, and it's like a hibernation. So cells will go through G1, S, G2, and M, and they'll reach this point somewhere, now we believe early in G1, where they'll make a decision whether to continue through the cell cycle or not. And if the conditions are not favorable, let's say no mitogens, the cells are packed against each other, they'll just sit there and hang out in G0. And they can sit there for a year, okay? They can hang out for very long times. And, but then when the signals change, a mitogen shows up, or they're not contact inhibited, they'll go through the cycle, come around again, hit this period, and again say, should I go through or should I hang out? And they'll sit and wait. Okay, that's what normal cells do. In cancer, in its essence, you could say, is that they just ignore G0. They just don't care. So instead of coming to M and saying, should I wait? They just keep going. They just keep growing and growing and growing. And in its simplest term, that's how we can understand cancer, that the cell cycle just keeps going and ignores G0. So at G0, the cell cycle stopped as well. And remember, we talked about EIF2. They also downregulate the protein synthesis. So it's a hibernation where they're basically just hanging out. And the cancer cells aren't going to do that, and they're just going to keep going and going and going. So what causes that to happen? Why do they ignore G0? Well, the answer is that a lot of things that we've already talked about can impact on that pathway, because the mitogens work through things like tyrosine kinase receptors, G protein receptors, the different mitogens. They can work through G proteins. They can work through membrane-associated tyrosine kinases, cytoplasmic tyrosine, threonine kinases, nuclear factors. Anything, as we'll see in more detail, that feeds into growth can regulate this process, either as a tumor suppressor or a proto-oncogene. So it's really the basic biology that we've already looked at. So I'm not going to go through, we're going to go through, periodically. I'm just going to give you some examples. But let me just give you a couple real-world examples of things that can happen. So remember how I told you, like with the tyrosine kinase receptors, you have two different receptors, and they're apart from each other. They don't transphosphorylate. Only if a signal, like a mitogen, comes will they come together and transphosphorylate. People can acquire mutations, one of these gene fusions, where a piece of protein that likes to dimerize gets put on the N-terminus in place of the normal mitogen binding thing. So what happens is you get these weird receptors that have a dimerization domain on the outside coupled to a cytoplasmic inside. And so what happens, because these pieces like to dimerize, even in the absence of a signal, a mitogen, the receptors come together, they transphosphorylate and activate the downstream cascade. It's a rare event, but it's exactly the kind of thing people find in cancer. So even though the mitogen isn't present, the cell on the inside thinks the mitogen is there, so it just keeps blitzing through G0 because it's fooled into thinking that the mitogen is there. Another example, very similar, is that in this case, the receptor somewhere in the membrane region undergoes some sort of point mutation that causes the receptors to stick together even though they're not normally, there's no signal there. So even in the absence of a signal, the receptors come together, transphosphorylate, and then send a signal to the inside. So again, even in the absence of a mitogen, the cell still thinks that there's a mitogen out there, and it just keeps going through G0. So these are examples of proto-oncogenic mutations in receptors. Now another one that's very related, another proto-oncogene is RAS. Remember we talked about RAS, this should be familiar to you. It switches back and forth between the GDP inactive and the GTP active state. Now RAS so what will normally happen with RAS is it'll be activated by a mitogen, very common, and then it turns on for a short amount of time, and then a gap comes along and makes it inactive. So only when the mitogen is there does the cell get the signal to grow. But RAS can undergo a very specific mutation only at amino acid 12, very specific in the whole protein, that one spot, that causes it to be unable or to hydrolyze its GTP to GDP even when the gap is there. Okay, so it stays active. And RAS is actually found in a huge number of human cancers. Somewhere over 25% of human cancers have a mutation in RAS. So what happens is even though the mitogen has gone away, the inside of the cell thinks that the mitogen is still there because RAS keeps activating the MAP kinase pathway, which keeps the cell going through G0. Okay? So RAS is a proto-oncogene because a rare mutation in one of your two RASs can cause the pathway to be active. Now, another thing that can happen is you can lose both copies of the RAS gap. So even though the RAS is normal, if you lose both copies of the gap in the same cell, the RAS can't hydrolyze its GTP to GDP. So the gap is a tumor suppressor because only if you inactivate both of them will it go on to lead cancer. RAS is a proto-oncogene, the gap is a tumor suppressor. And this is a theme we'll see over and over again, that within the same pathway, we're going to see both proto-oncogenes and tumor suppressors. So I'm going to leave it here, and we'll get into RB, which is one of the classic tumor suppressors. We'll go through that on Wednesday.